In this video, we'll have a look at chromosomes, talk about heredity, and also start to define some terms when it comes to genetics. So let's start off with some of the most basic terms, and that's heredity. And that's basically the transmission of genetic characteristics from parent to offspring. Now here's a term I believe we've discussed before, and it is a karyotype. A karyotype is a way for us to have a look at the complete set of chromosomes within an individual. So you harvest the cells, take blood sample, tissue sample, you grow the cells, and then you prepare them in such a way that the chromosomes are in basically a metaphase type look, and then you lay them out in order. So it's basically a constructed chart of all 46 chromosomes laid out in order of size. And there's 46 chromosomes, yet there's 23 pair of homologous pairs. Now, then the question becomes, well, what's homologous refer to? So homologous chromosomes, this is where one chromosome in the pair is inherited from the mother and one from the father. And each one of these homologous chromosomes will have corresponding alleles or genetic features on it that it'll code for. 22 of the pairs will refer to as autosomes, and one of the pairs will be the sex chromosomes. And of course, this is the X and the Y chromosomes. This is a picture of a karyotype, and once again you can see that we have chromosomes and we've given them numbers 1, 2, 3 all the way down to 22, and you can see within a given pair we're looking at homologous chromosomes. And as I said, homologous chromosomes suggests that they have the same alleles or coding regions on them for same traits. And here we have 1, 2 pairs, let's move over here, pairs 1 to 22 are the autosomes, and the X and the Y are considered the sex chromosomes. Now, another term to be familiar with is that of diploid, also known as 2N. And this describes any cell that has 23 pairs of chromosomes. And somatic cells typically are diploid. There's a few little exceptions, but in general we can assume that they're diploid. And diploid simply means that they have both pairs. Both of the chromosomes are there within the pair. Haploid it describes cells containing half as many chromosomes. And that means one of the partner within the pair is not there. And of course, we see this when we're looking at germ cells. The production of sperm or egg through meiosis. It's basically a reduction divisions to go from a diploid to a haploid. So when a sperm and egg come together, then they reestablish a diploid number in a zygote. Another term is locus or loci, and this is the location for a particular gene on a chromosome. It's basically its address. Alleles are different forms of a particular gene. Now, alleles are found at the same locus on homologous chromosomes. And of course, here's some other terms you've come across before. You can have dominant alleles. And typically, when we write out dominant alleles, we want to say it's the dominant version. We use a capital letter. And that's versus recessive alleles, where we'll use lowercase letters. Well, what do these terms mean? Is that if for dominant, if present, the corresponding trait is usually seen in the individual. And it typically masks the effect of the recessive trait. And typically this is often because this gene produces a protein that has a role in that visible trait or the phenotype that's expressed by an individual. Now for recessive alleles, as I said, they're represented by a lower letter. The corresponding trait typically is only seen when the recessive allele is present on both homologous chromosomes. Now, genotype. This is the alleles an individual possesses for a particular gene. And since you have two pairs or two homologous chromosomes, the alleles don't have to be exactly the same and code in the exact same way for a given phenotype. But if I have a homologous individual, they have two identical alleles for the gene. Heterozygous, so it's homozygous, I should say, versus homologous. Heterozygous individuals have different alleles for that gene. Now, I've used the word already. I mentioned it once or twice already, but let's define it. A phenotype. The phenotype is the trait you can actually see. It's the observable trait. An allele is expressed if it shows a phenotype for an individual. Now, we're not going to spend much time here. Many of you have probably seen this in a lower biology course, the concept of a Punnett square, where you can write out look at recessive versus alleles by forming a little square with four corners or more in it, depending on the nature of the genes you want to look at. And we're not going to spend much time. This is sort of what a Punnett square would give you. It looks at the different allele combinations. Now, just a couple of other little terms is that I just want to mention this, a career that many people don't actually think of, and just take a second to mention, is genetic counseling. It's a medical profession, and what a genetic counselor does is they perform genetic testing, and often they act as an expert to allow, say, patients or couples understand the consequences of having certain groups of alleles within them in terms of, say, potential risk for certain diseases or risk to potential offspring. Now, often when we're talking about alleles, often one gene does not act by itself. It's influenced by the environment potentially, or it works with a series of other genes to create certain features or a phenotype within the body. The gene pool is the genetic makeup of the whole population. Multiple alleles, more than two allelic forms of a gene. Now, here's an example we haven't gone into, but in 1220 we'll come back to, and that would be looking at the blood typing system in humans, where you have the ABO system, where there's multiple allelic forms for that gene. You may hear the term codominance. In this case, both alleles are equally dominant, that both get expressed. So it's unlike the recessive versus dominant alleles we talked before, where a dominant one, if it's present, will get expressed, and for recessive, both of the homologous chromosomes have to express it for the recessive trait to be seen. In codominance, what you have is both of the alleles, the different alleles being expressed. And an example that would be the blood system, but we haven't done it, so we're not going to worry about the details of that particular system. Incomplete dominance is when a heterozygous individual shows a phenotype somewhere between the two traits. Between the traits each allele would have produced. So in this case, it's sort of a halfway meeting between the two traits that could be produced by the two alleles. Now, polygenetic inheritance, that simply means, look at the poly means many, genes at two or more loci is location, contribute to a single trait. So something like eye color is not based on one gene. It's a number of genes that interact to produce eye color in an individual. Pleiotropy, one gene can produce multiple phenotypic effects. Now let's have a quick look. So here's polygenetic inheritance. So in this case, we actually need 3 genes to work together to produce eye color. In pleiotropy, in this case, one gene can actually lead to a number of different types of phenotypes. And once again, we're not going to worry about this particular example because we haven't discussed any of the pathophysiology of it. Now, you may hear the term sex-linked, and these are alleles that determine often phenotypes that are carried on the X or the Y chromosome. And in this case, because certainly if you think about it, females typically we have XX, and males we have XY chromosome. That any allele variation, or especially if there's an issue with developing phenotype from it, tend to be inherited by one gender or sex more than the other. And some examples would be certain types of color blindness. But once again, we haven't done those examples, so we won't dwell on them. I think it's more important that you know what the basic definition is for these terms. The term penetrance of an allele, it's a percentage of the population exhibiting the expected phenotype. Now, I want to point out that environmental factors may actually influence gene expression. So there's often this discussion, nature versus nurture. Is it your genes or is it in the environment? And in many cases, it's an interaction of both of them. Now, I just want to say, because we had talked about dominant and recessive alleles, at the population level, it's important to know that that often people have the wrong impression that a dominant allele makes it sound like it's much more common than the recessive. And that's not necessarily the case. Dominant and recessive alleles just talk about their influence on a phenotype, not the percentage of the population that has that given allele. Some examples are polydactylia. Polydactyly refers to extra fingers or toes. And we can also see that in blood grouping. But once again, as I said, we haven't really done that, so we're not going to spend much time. Epigenetics. I did post a video about epigenetics as an FYI video. I strongly encourage you go have a look at it. It's a fascinating field. And really, it's a field that looks at non-genetic factors that can alter gene expression. And often this altered gene expression can get passed on to offspring. And this is a fundamental rethinking of the way we used to think about genes, that the only way they changed was through mutation. But now we know these things that sit on top of the DNA, the epigenetic factors, have a large role in controlling the function of the DNA. And these can be shuffled, they can be arranged, they can be changed. So we can have gene expression being changed without genetic mutation to the base sequences. Now, one of the most common ways that we control this are the epigenetic tags, and one of them is DNA methylation. And this sounds familiar because it involves a methyl group. And in this case, we can change the methyl groups. They can be added or subtracted from the DNA, and that affects its expression. And often the methylation will silence genes, basically tell them not to be turned on or copied. And certainly, we now know that inappropriate DNA methylation has a role in some types of cancers. Now, we're just going to say a little bit about cancers. It's a huge subject that touches many of us in our lives. But often when we're talking about cancers, we're talking about malignant tumors. And that is the basis between many cancers. And of course, the hallmarks of them is that they tend to have uncontrolled growth. They tend to metastasize. Metastasize means they move from their primary site to other sites where other tumors may develop. And this is different than benign tumors. And benign tumors typically are slow growing or really hardly ever growing that much. And they're often encapsulated. And that's one of the hallmarks, that they actually have some type of capsule around them. Now, oncology is a medical specialty dealing with the treatment, diagnosis of metastatic or disease or tumors. Tumor angiogenesis is an interesting phenomenon, and that is the growth of blood vessels by energy-hungry tumors. There's this phenomenon that tumors will often secrete substances which will recruit blood vessels to them. And cancers are often named for the tissue of origin. Carcinomas from epithelial tissue, lymphomas from lymph nodes, melanomas from the pigmented melanocytes in the epidermis, leukemias from blood-forming tissue, or sarcomas, which refer to bone or other connective tissues or muscle. Now, carcinogens are environmental cancer-causing agents, and it's believed that only about 5 to 10% of cancers are really rooted in heredity. And a lot of them are probably a result of multifactorial factors, including environment. And of course, some of the most common things that could cause gene mutation would be radiation, like ultraviolet rays, X-rays, chemicals. Certainly cigarette smoke is a major cause. And viruses like the human papillomaviruses, hepatitis C, and herpes simplex 2 viruses. Now, oncogenes cause cell division to accelerate out of control. Now, onco, of course, refers to cancer, and it may seem odd that we have these things called oncogenes, but typically these are most likely, in most cases, genes that deal with normal cell division that have gone out of control. And typically what they do is they produce excessive amounts of growth factors or growth factor receptors, and that helps to stimulate mitosis. There are tumor suppressor genes, and healthy tumor suppressor genes really help to inhibit the development of the cancer. And some of them actually code for DNA repair enzymes, but of course, because often cancer is that the DNA has become mutated, so if you can repair it, you may be able to set it back on the right course. Now, of course, if you have mutated tumor suppressor or silenced tumor suppressor genes, then it allows for the oncogenes to act unopposed. And of course, what are the effects of cancer? Well, one of it is that you actually replace functional tissue with non-functional tissue. Also, they're very heavy on the use of nutrients. So it really pulls nutrients from other tissues, and which then have an effect on those tissues. Or you can have a weakening of one's immune system. And often it opens the door for opportunistic infections. Opportunistic infections are those infections that typically don't occur unless conditions have been set up for them to occur through things like immunosuppression. Now, we do know that many cancers actually have ways to evade the immune system or to turn off parts of the immune system. And that's the end of this chapter.